Okay, well, welcome to the video number 3 for week 1. Um, let's get right in. We're going to talk about the process of a statistical study. Your objectives to describe what the process is and identify some various types of studies. Again, a lot of this is going to be some initial vocabulary and concepts around that. Um, so to conduct a statistical study, basically we start with determining the design of the study. What is the question we want to study? What is the population we want to study? What are the variables we need to use? And how are we going to sample that population? This is all part of the designing things portion. Then, once we've determined what variables we're going to measure and how we are going to sample those, what measurements we're going to take, um, we collect the data, then you organize the data, then you analyze the data. Um, Better job you do up front here in step 1 in determining your sampling method and what variables you want to measure, the better your results are going to be. You can mess anything up in any step here, but getting step 1 right will help a lot with getting a good study. Okay, so let's do our example here where I want to identify the population and the variables. So bunch of neurologists want to study the effect of vitamin C on nerve disorders. The goal was to see if taking an intravenous dose, so injecting it into a vein, if an intravenous dose of vitamin C will reduce the amount of nerve pain reported by patients. Um, so we want to identify the population of interest and the variables in this study. Go ahead and take a minute, think about what those could be. We'll come back and see, pause, take a minute, and then move on when you're ready. So, right, we're looking to see if a new treatment will reduce nerve pain in patients with nerve disorders. So our population is patients with those specific disorders that cause some form of nerve pain. Most likely, it'll be a little bit more narrow, probably to a specific type of nerve disorder. Rather than broadly any nerve disorder that might fit. Variables of interest are the amount of vitamin C administered and the amount of nerve pain each patient reports. Okay, so there are broadly two types of studies you can do. You can do an observational study where you're looking at data that already exists, or you can do an experiment where you are generating data to identify cause and effect relationships. The example we were just thinking about a minute ago is an example of an experimental study. An observational study would be using, for example, looking at weather reports in Marietta for the last 10 years to see if the amount of sunlight has, you know, in per month has changed or something like that. Um, okay, so for each of these two things, would we look at an observational study or an experiment? So if we wanted to determine the average age of college students across the nation, would that be an observational study or an experiment? If we wanted to determine if flu shots help prevent severe cases of the flu, would that be an observational study or an experiment? All right, take a moment, think about it, unpause when you're ready to move on. Okay, so the first case would be an observational study. We just need to look at the existing records of college students and get an average age from those. The second case would be an experiment where after a cause and effect relationship between flu shots and severe flu prevention. Um, in observational studies, we usually want to look at what's called a representative sample. So we want to pick a sample of the larger population that has the same relevant characteristics as the whole population and doesn't favor one group over another. So in the college student average age, it is going to be somewhat difficult to gather the records for every college student in the nation. You'd have to get cooperation from every college in the nation, permission to access the student records, etc., etc. Um, so you're going to have to pick a sample of college students and base your results off of that, right? So the thing is, we want a sample that's going to be similar in characteristics to the overall population. So we wouldn't, for example, go to an all-girls college, if those still exist, um, and base it solely off that, because that might have different population characteristics. It would be favoring one group within the population over others. We wouldn't want to base our sample solely off of incoming freshmen, because again, that would skew the results quite a bit, because we'd be favoring the youngest, what's likely the youngest portion of the population over the others. So there's some techniques you can use for sampling. You can take a random sample. You can do that very simply. You can take a stratified sample, a cluster sample, systematic sample, or a convenient sample. So a random sample, everyone has an equal chance of being selected. So the bingo machine ball is a perfect example. Um, Hopefully all the balls in that are the same weight and have equal likelihood of being selected, as long as nobody's doing anything screwy. Right? You spin it, they're mixed, they're random, you reach in, you grab one, every ball has an equal chance to being selected. The simple random sample, every sample from the population has an equal chance of being selected. This is probably best understood by comparison with some other schemes. So let's look at the next one. Have a stratified sample. Divide the population into subgroups called strata, and the same number of subjects are chosen from each strata. So, for example, in the college age, you'd select the same number of first year, second year, third year, and senior students. Skews things a little in terms of in each compared to a very simple random sample, each potential sample does not have the same likelihood, because we're taking a subsample from different equal amounts. Our sample consists of equal amounts of different groups. So the likelihood that I get a sample that's heavy in first year students is gone. So it's no longer a simple random sample. I'm still randomly sampling within each strata, but within a larger scheme. A cluster sample is another one. We divide this population into subgroups called clusters. Each cluster needs to be similar to the population, and we pick whole clusters at random. So a field like this would be a good example. If you divide the field into little zones, if you randomly select zones, you've got this cluster of individual rice plants in each zone that should be similar to any other rice plant in that field. A systematic sample, you're selecting every however member of the population. This is, as you can see in the diagram there, fairly common in production type environments. You're selecting every, maybe you're selecting a sample of French fries every hour. Maybe it's every fourth bottle. Maybe it's every 10th widget. whatever it is, every so often you collect a sample on a scheduled basis, systematic. Finally, you've got convenient samples, and that's what is convenient for the researcher to select. So if I was, for example, if I were to do a study about the attitudes of college students at WSCC, a convenient sample would be you guys in the class. That would be a convenient sample for me to take. All right, so let's look at an example. We've got 3 situations. Which sampling method is... All right, we've got 5 situations. So let's take these, take a minute, get these 3, pause it, put down what type of sampling is used in each of these 3. We'll move on to the next slide, pause it, come up with your answers, then we'll move on. Okay? So if we're surveying 50 people in each of a senator's 12 voting precincts, in number 1, in A, and B, we're sampling every 10th box off the assembly line. C, we've got a student who's walks down the halls in her dorms to ask her fellow students a question. Okay. All right. And D, we've got 5 school districts in Chicago. We chose 5 out of however many school districts are in the Chicago area, and we're asking each household in each of those districts about the school-age children at home. Finally, we've got a $100,000 shopping spree based on a name draw out of a box of entries. All right, so pause, move on when you're ready. So A is an example of stratified sampling. We've got 50 voting precincts, or is it 12 voting precincts? But each precinct is the strata. We take the same number of data points in each strata. B is systematic sampling. We're taking every 10th box for testing. C is convenient sampling. It's just, whoever happens to be in the dorms at the time. C is cluster sampling. We've got the 5 school districts. Each school district represents a cluster that should hopefully be similar to the overall population of all the school districts in the greater Chicago area. Finally, it's random sampling. Every name in the box should have an equal chance of being chosen. Okay. Types of observational studies. There's cross-sectional, where you get the data at a single point in time, and longitudinal, where you gather data at multiple points of time over a long period of time. So let's look at the example. And again, when you're done, when we get through this, pause, make sure you've got your idea set, and unpause when you're ready to move on. So if we've got 220 patients, we follow them for 15 years to find the long-term effects of gastric bypass surgery. And in B, we've got a gastroenterologist surveying 130 of his patients 6 months after their bypass to see how much the average weight loss is for his patients. Okay, so in the first one, we're following the patients for a long period of time. This is going to be a longitudinal study. And the second one, we're looking at everything at a specific point in time, so it's a cross-sectional study. Some other broad types, there's a meta-analysis, where you're analyzing the results from previous studies and coming up with an overall conclusion based on a bunch of previous studies. And there's a case study where you look at multiple variables that could affect a single event, and you look at that single event in more depth. Okay, so let's classify these 2 examples as either a meta-analysis or a case study. So First, we've got oceanographers studying research on tsunamis dating from 1900 to 2000 to determine their effects on the ocean floor. Is this a meta-analysis or a case study? And the next one, meteorologists studied the Indian Ocean tsunami of December 2004 to try to identify warning signs. Meta-analysis or case study. All right, the first one, we're looking at multiple events. We're looking at existing data. We're pulling things in from studies and combining them and coming up with an overall effect of the ocean floor. So this is a meta-analysis. The second one, we are looking in a lot more depth at a single event, studying multiple aspects of it. So it's a case study. Okay, let's move on to experiments. Okay, so some basic definitions. A treatment is a condition that is applied to a group of subjects in an experiment. So it's what's being done. So in one of the examples we talked about a few minutes ago, they were giving people vitamin C intravenously. So that would be our treatment. Subjects are the people or things being studied. So, the people getting the vitamin C would be a subject. If we were changing something in the production line and following that systematic sampling thing, the subjects could be at that every 10th box of cereal to see how long it took the change we made to go through something. So subjects could be people or things. Participants are specific to people. They are participating in your study. They had better be participating willingly. You get in trouble for not doing that. So the response variable is whatever responds to the treatment. So, for example, in the vitamin C study, the response variable is their level of nerve pain. The explanatory variable is the variable that should cause the change. So it's the amount of vitamin C there that you're injecting. So some principles for experimental design is you need to randomize who's in what group. You're going to have a control group and a treatment group. One group gets the whatever the treatment is, and one group does not. So you can measure what the effect of the treatment is in comparison to not having it. You need to try to control for outside effects on the response variable, and you need to replicate it to enough times that you can tell whether or not there are meaningful patterns. And a lot of that is why drug trials take so long, because all of this has to happen and you have to do it enough times that you can really see. So, as I just mentioned, a control group is a group which does not get the treatment. The treatment group is the group that does get the treatment. Confounding variables are any factors other than the treatment that cause an effect on the subjects of the experiment. So with the vitamin C and the nerve pain, one of the reasons you might look at just a single nerve disorder that could cause nerve pain, it would be that the different disorders could have different mechanisms, and vitamin C might impact people with one disorder more than another. So having multiple disorders in play would be a confounding variable. It would affect our results and not be related to the treatment. We've also got the placebo effect. You've probably heard about this. I think sugar pills. So it's the response due to the power of suggestion rather than the treatment itself. So it's a substance that looks like the actual treatment but contains no intrinsic beneficial things. Hence sugar pills. So make a pill that looks the same as whatever you're using. Inject saline instead of vitamin C. Do something that mimics the appearance of the treatment without actually being your treatment. Um, We've also got single blind and double blind experiments. So in a single blind experiment, none of the subjects know if they're in the control group or the treatment group. But the people running the experiment know which group is which. So when I was at Washington State University, there was a food science department. They would occasionally have taste testing experiments. So you could go in and you'd get a free ice cream cone from the creamery down the hill, um, and taste a few things and record your observations. And as a participant in that, I never knew what I was tasting. It was just one was different flavors of yogurt, and I didn't know which was which, and so it was single blind. The person giving it to me, I believe, knew which was which. Um, in a double blind experiment, the people interacting with the subjects also don't know which is which. So you'd have somebody somewhere who kept track of which bottle, which group got the placebo and which group got the actual medicine, but the people actually dispensing it and taking the measurements and stuff and interacting with the subjects um, would have no clue. So the idea is that there would be no chance of them biasing the results somehow. Okay, so let's go back to our vitamin C example. So we've got it narrowed down to only patients with multiple sclerosis. We get 40 participants who have MS with nerve pain. We have them split into two groups. Group A gets the vitamin C, so they're our treatment group. Group B gets saline, so they're our control group. It's no active ingredients in saline, it's just highly sterile salt water. You track their pain levels. The patients are not told which group they're in, but the nurses giving the IVs are aware of the group assignments. After some length of time, the pain reports are compared to determine if vitamin C reduces the amount of nerve pain. Okay, so what are our explanatory and response variables? What's our treatment? Which group is the treatment group and which is the control group? I think I already gave that away. What's the point of giving saline to group B? Was this a single blind or double blind study, and do you think this is the best choice? Okay, so since the explanatory variable is what explains the changes in the response, we want to know if vitamin C can reduce nerve pain, so our explanatory variable is the dose of vitamin C, as we discussed several slides ago. The response variable is then the amount of nerve pain. Treatment is what's being applied to the group, so the treatment is the dose of vitamin C. Group A received the treatment, so it's the treatment group. Group B is the control group because it did not receive the treatment. Our saline is the placebo. It's administered to compensate for the placebo effect so that they get the same suggestion that they're receiving treatment. It's a single blind study because the nurses know which group is which. Pain is a subjective measure, so it could be easy for the nurses to unintentionally influence the responses. So a double-blind experiment would probably be a better choice. Last thing in this portion is institutional review boards. So this is a group of people at the institution. They review the design of the study to make sure that it's appropriate, that there's no unnecessary harm, that that there is a plan in place for any harm that does occur, that your consent procedures are in place so that participants know what your study is about and what your goals are. If you're going to be deceiving your participants for any reason, that has to be explained and examined and gone over. I have, since I was doing educational research, I have submitted things to the institutional review boards before. It's even with an expedited or exempt study, it is still a process. They examine things fairly thoroughly. So if you are going to be doing research with living subjects of any sort, it gets reviewed. All right. So let's take a break for a bit before you come back for the fourth video.